Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gill Eapen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gill@eapen.info. My guest today is Professor Debby Silver, who's Associate Professor of Molecular Genetics and Microbiology at Duke University. Her lab studies embryonic brain development. Welcome, Debby.
Thank you. Thank you for having me.
Yeah, thanks for doing this. So I want to start with one of your papers, Dynamic mRNA Transport and Local Translation in Radial Glial Progenitors of the Developing Brain. You say in the developing brain, neurons are produced from neural stem cells termed Radioglia. Is glia the right pronunciation for it?
Yeah, most people actually refer to it as glia, but there is no right or wrong.
Yeah, so stem cells termed Radioglia. And you say radial glial progenitors span the neuroepithelium, extending long basal processes to form end feet hundreds of micrometers away from the soma. So before we get into those details, Debby, I want to get some sort of definitions for these things. So end feet is really the end of the neuron. What exactly is it?
Sure. So the radioglia are these just really fascinating, amazing neural precursors, actually. So they're the cells that contain the end feet, and these are the cells that will generate neurons. And they were kind of historically observed to be be elongated, hence their name Radial, and to bear features similar to glial cells. But what was subsequently found over the years was that they have several functions. One is to provide us almost a ladder by which new neurons can be make their way up into the brain. And the other is to generate neurons. And so these the end feet structures that you were referring to are simply the structures at the very end of these radial glial progenitors. There are end feet at both the very bottom of the developing cerebral cortex. These are referred to as apical end feet. And then there are end feet at the very top referred to as basal end feet.
So they're produced somewhere. I'm just trying to abstract it a bit. So they're produced somewhere in the brain, and do they migrate from their site of production to somewhere else?
So the radioglia themselves are going to span the cerebral cortex wall. And so basically our brains have ventricles but during development, which are quite large. And these ventricles, so the apical, one part of the radioglia is going to be at the bottom, one part's going to be at the top. And themselves, at the stages at which they're making neurons, the radioglia do not migrate far. But the neurons that they migrate, the neurons that they produce do migrate up, and they basically go in what's referred to as a radial pattern. So it's as if they're moving from the very bottom where the ventricle is located, all the way up into what is referred to as the cortical plate. And this distance is going to vary depending upon the stage of development that you look at, as well as the organism. So it could be several hundred micrometers by which they're migrating, or even centimeters in a human brain.
Yeah, it's just a fascinating thing to think about. So all of this activity is programmatic, and it sort of happens, right? It's it's not something that that it's some external force is doing it, right? It's all programmatic at the beginning.
Yeah, that's right. I mean, there's somewhat stereotypical modes by which neurons will make their way into this this part of the developing brain, and we know features that they have, morphological. We know about gene expression that is associated with migrating neurons, but the what actually directs it is still kind of territory that a lot of scientists are trying to understand, and the extent to which this is pre-programmed versus whether these neurons, as they're migrating. I think another perspective is that the neurons, as they're migrating, are encountering different milieu, if you will. So they're encountering different cell types that themselves could help to redirect or direct a neuron to its proper location.
Well, yeah, so if it doesn't reach its intended location, then that is going to result in some sort of disease, right? Something that's going to have long-term.
Yes, correct. So there are a number of neurodevelopmental pathologies that have been linked to deficits in the ability of these neurons to find their right position, their final position. There are disorders that are linked to patients having intellectual disability, as well as abnormal malformations of the brain. And so these can really vary a lot, but neuronal migration is really essential for proper development of the brain. And so there are a number of diseases that are linked to it.
Yeah, is this specific to the human brain, or this happens in any non-human animal too?
Yeah, so that's a great question. In fact, my lab, as well as many others, takes advantage of model organisms to study this process, in large part because many of these processes are highly conserved. That is, neuronal migration has to occur in human brains as much as it has to occur in non-human primates, or even in mouse models, which is what we heavily study. And what's also quite fascinating is you can even reproduce some of these events by just simply regenerating a tissue in a dish. So there's now scientists who are taking cells and using them to make mini neurosphere structures that are similar to share features of a developing brain. So there is not only a lot of evolutionary conservation, but there's that just tells you how important the event is that you're able to reconstruct it with the tissue.
Mm. And the human brain obviously is developing for a long time, but early on the development is quite dramatic. So the patterns here, I guess two questions: are they sort of measurable? Number one, and number two, if they're measurable, could we actually use those patterns to predict some sort of disease state that it might end up in?
So ultimately, what will end up happening at the kind of the end stage of this process of neuron production, which is referred to as neurogenesis, ultimately what's going to happen is that the neurons are going to find their kind of final location, if you will, in the brain. And that itself is actually pretty predicted. You can predict that. You can predict not only where those neurons are going to reside, but in fact, there is a pretty striking correlation between the position that those neurons ultimately land in and when they were born. And so that in itself is pretty predictive. Now, remind me what your second question was. I apologize.
Yeah, so I was wondering if we can sort of observe what's happening in the brain, without any intervention. We can just observe. Could we actually potentially predict some sort of disease that might occur in the future?
Well, I think the short answer would be potentially yes. I would say that something, there's a lot of events that are happening simultaneously. So neuronal migration deficits have been associated with a number of disorders. Now, in that regard, you might say, well, you can make a prediction. If you knew that there was a neuronal migration defect, maybe it would suggest that this particular disorder is more likely. And again, I want to caution and say that these are mainly based on studies that have been done in model organisms.
Right.
But the other important aspect of this is that a lot of these events are happening simultaneously with other processes, like neurons being made, neurons beginning to form connections, et cetera. And so, it's important to find ways that we can, understand cause and effect and disentangle different processes.
Right. Yeah. So you mentioned, Debbie, if I understood it correctly, that we can predict where the neurons will end up based on the time they were created. So time meaning during the progression of the brain.
That's right. I mean, this is a fascinating observation that was made many years ago, and it was done using a large part by studies that were done in non-human primates, as well as followed up in mice. And what these historical studies did really beautifully was to basically use these tricks to label cells at their birth date, and then to be able to follow them over time. And so essentially what one could do was to label a cell at one stage of development, and then look at this very endpoint of development and see where that cell that was labeled at that early stage landed. And then you could compare it to a cell in which you labeled its birth at a much later time in development. And so you're essentially providing a time stamp, if you will, on the neurons that were made at that time, and ultimately being able to evaluate where they land. And so that is a pretty uniform principle in mammals. Some of these rules get a little bit trickier in other species.
Right. Yeah, so it sounds like it's on a schedule of some sort, and it is sort of sequential. So if you disrupt, one aspect of it, does it have sort of a domino effect for the rest of the schedule?
Yeah, that's actually a fascinating question. I can't really answer that with a yes or no because it's a little more complicated. There is evidence that if you disrupt it, say the migration of one at one specific time, the brain will actually recover and realize that those neurons have not made their way to the right location, and will, in some cases, there's a process that's referred to as signaling, by which we think that the brain may be recognizing the absence of those neurons and actually telling these precursor cells to make more of the neurons that are missing. So that can happen, but there's also evidence that it could have a domino effect where disrupting the migration of one neuron will then lead to disruptions at other stages as well.
Yeah, so there's some sort of signaling back to the production center, so to speak, if they're not finding them at the location at the right time, so they can sort of restart the process.
Yeah, and I think, and this comes back to, this is an area that I think is absolutely fascinating in my research field, one that we're interested in, but some really fantastic labs are also working on to try to understand, how the developing brain has these checks and balances to recognize if cells are not in their right place, or maybe there are cells that are positioned there, but they're not the right types of cells. And so the idea then is that there may be signaling potentially back through these radial glia because they have this long structure, to inform these radial glial progenitors to say, okay, hey, we are missing this type of cell. You need to now change your directive and make the cells that we're missing.
Right. Yeah, it's fascinating. You talk about autism in this paper, the autism protein FMRP. And what's the connection?
So this is so the paper that you're describing is a paper in which the observation that we made is that there is localized gene expression within these highly elongated radioglial progenitors, and so we found that this could happen. That there were actually hundreds of RNAs. So normally what's thought in a cell is that the RNAs are made in the nucleus, or in the nervous system, we refer to this as a cell body. And these RNAs then are primarily functioning nearby the nucleus. But what has been discovered is that in highly polarized cells, they maintain a process whereby they can rapidly generate proteins from RNAs in a both spatial and temporal specific fashion. So imagine a cell that is really, really elongated. It's much more cost effective for that cell to make the proteins that it needs in this distal compartment, rather than shuttle the components it needs to make that protein, and shuttle the protein itself over far distances. So in our paper, we found that RNAs are highly localized in these distal compartments, and so we began looking, asking, well, what could be helping in this process of shuttling, and in this process of telling these RNAs to be made into proteins. And so to tackle this, we started thinking about, well, what are some factors that we know have known roles in controlling RNAs? And this is where we happened on this autism-related gene, FMRP. So this gene, as you mentioned, is heavily mutated actually in Fragile X. The mutations are which is one of the so mutations in FMRP will cause Fragile X syndrome, which is a form of related to autism, and FMRP itself is known to associate with RNA. So we found, in fact, that FMRP may be having this unappreciated role in helping to localize and direct the functions of this gene expression at the very top of the brain.
I remember, Debbie, I don't know the details of it. Something came through the news last week about a connection between Parkinson's and this sort of a mechanism. Did you see?
I did not, but it sounds quite interesting.
Yeah, it's something to do with, I don't know if it's the right term, sort of elongation and the velocity or the distance of transmission. And I think the paper is talking about, the connection between that and possibly Parkinson's.
Well, this process that I just described to you, whereby a cell that's highly elongated uses local gene expression to control its function, is one that is really predominant, thought to play predominant roles in neurons. And so we've been looking at it in the precursors of the neuron, but it also is really well known for its role in a number of functions of neurons. And so in this regard, it doesn't surprise me so much that there may be roles for RNA regulation in something like Parkinson's. We know that RNA binding proteins like FMRP are heavily linked to other neurodegenerative disorders.
Right. Yeah. So the brain, once it's sort of settled, it's not that plastic, right? You can't really restart some of these processes once they're settled.
Well, there is evidence that certain parts of the brain can regenerate neurons as they're needed. There's evidence that this can happen in the hippocampus, although this has been challenged more recently. And so I think there is some debate as to the extent to which this happens in humans. But, there is some plasticity to the brain in terms of its ability to respond to injury and not only regenerate neurons, but also redirect cells to the right place. And there's some really nice work that has been done where people can take neurons and actually put them into a brain that has been injured. And in some cases, those neurons are able to reform the connections that had been impaired.
Right. I know that we are not there yet, but do you if you look forward, do you see possibilities for us to do it artificially with artificial neurons when there is something is wrong?
Well, wouldn't that be great? I think there is a lot of amazing research that's being done to understand how neurons are generated, to understand how to make neurons even in a dish, and how once you make those neurons, you can tell it to functionally integrate with other neurons through synapses. And so as those foundations are kind of laid through basic science research, I think the possibility is potentially there. I mean, obviously, technically speaking, putting a neuron into a developing brain is not anything that we're anywhere near being able to do. But people have done experiments using model organisms, and there does seem to be a lot of plasticity both to the cells that you put in as well as to the environment that you put them in.
And you mentioned, Debbie, the regeneration capabilities are different for different parts of the brain, right? There's certain places where it's more compared to others.
Yeah, that's right. So it's certainly gonna vary across the brain, and that's something that we've been limited in our ability to understand these processes in large part to model systems where we can study it in, say, a mouse brain, or study it in some non-human primates. And I say we as a field. Being able to then make these connections and inferences as to what's happening in a developing human brain is a whole nother question.
Yeah, but the mouse brain actually gives us sufficient information to make some hypotheses as to what a human brain is.
So there's a lot we can learn from studying the mouse. We know that there's a lot of genes that a lot of the genes in the mouse are also acting in the same processes in human and vice versa. And so I would say there's a lot of important information we can glean, but there are processes that are going to be different. The most notable, for example, humans, of course, have folds in our brain, and mice have lissencephalic brains. They're smooth. So if you want to understand the basis for something like gyrencephaly, which is folding, the folds of the brain, you studying it in a mouse model may not be the best way. We also know that there are important differences. So the humans have certain genes that are produced in humans that are actually not found in the mouse genome at all. And this has led to some really fascinating evolutionary innovations where we've acquired potentially new cell types in the developing human brain that are not present in a species like mouse.
If you're interested in the folds, what would be the next best model? Is it do we have to go to chimps or something like that? Or we go further down?
You can actually go further down, and there's some really amazing work that has been done in a mammal called the ferret. So I have some fantastic colleagues in this field who have been using ferrets to better understand abnormal folds that can form, and also, to model diseases that are associated with abnormal gyral patterns.
Yeah. Excellent. Yeah, we'll take a quick break, Debbie. When we come back, we'll talk about a couple of your other papers. Thanks. This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com. Debbie, we were talking about the development of the brain, neurons, how they're produced, how they might transport themselves to different parts of the brain, and what that implies. You have another paper entitled Prolonged Mitosis of Neural Progenitors Alters Cell Fate in the Developing Brain. You say embryonic neocortical development depends on balanced production of progenitors and neurons, and the genetic mutations disrupting progenitor mitosis frequently impair neurogenesis. So mitosis is cell division, isn't it?
It is, and so broadly speaking, it's the process whereby one cell can divide into two. Now, aficionados will actually refer to this as some people will refer to it as a cycling cell, but we actually will refer to it as that actual stage at which a cell is about to complete its production of two new cells.
Okay, and so what's the crux of the paper here? So before we get into it, so what's the difference between progenitors and neurons?
Great question. So the progenitors is, you can think of it as sort of the mother that's going to make the neuron. And so a progenitor here is a more specialized type of stem cell. So a stem cell is any cell that can undergo division to make more of itself, as well as make a more specialized cell like a neuron. And so there are populations of these neural precursors in our brain that we refer to as neural stem cells.
Okay, so prolonged mitosis implies it just taking longer for that process to complete?
Yeah, that's right. So in this paper, what we found is that if you look at genetic mutations that what we discovered was that there were genetic mutations that would basically cause these precursors to take a much longer time to divide. And as a result, this was associated with alterations in the in the types of cells that were made. And then we wanted to take that one step further and ask, well, if we specifically perturb this mitosis and using independent methodologies, can we recapitulate this outcome? And the short answer was yes.
And so you mentioned here sort of a mitotic duration. So we have an expectation of how long this is supposed to take.
Yeah. So during development of the brain, these precursors undergo a pretty stereotyped timing in which they will basically take a certain amount of time to go through different types of changes in the cell cycle, and then to complete this process of mitosis. Now, this timing of mitosis and this duration can change in a normally developing brain as the brain gets proceeds through development. So the earliest stages, it's going to be quicker than at the later stages. And what is, I think, really fascinating and was probably underappreciated was this idea that the duration of mitosis specifically could have an impact on whether a neuron is made or not. Prior to our study, there was some really nice work that had been done by Colette DeHay and Wieland Hüttner's groups. They had shown that other stages of the cell cycle of progenitors can influence their the generation of neurons, and they had not looked specifically at this stage of mitosis.
So the neuron generation that is driven by the mitosis of the progenitors, is that duration mechanistic, meaning is it the same all through the development of the brain, or it changes as time passes?
So there is going to be some differences in the duration if you look at a younger stage of development versus a stage just towards the very end of development prior to birth. Earlier on, it's going to be slightly shorter, and then later it's going to increase in its length. And this has been something that has been observed for a long time, but it's not entirely clear why that is the case. Why the cells change their duration over time.
And so presumably there's some correlation, right, between those. So I'm thinking back to our earlier conversation. If we sort of study the progression, could we pick up something that is not working as expected?
Yeah, potentially. I think that's one of the take-home messages that that we would like to have proposed from our paper. If there is an abnormal duration of cell division, it might be telling you about problems that can arise. So there were two outcomes that we observed that were associated with this lengthening of cell division or lengthening of mitosis. The first problem was the cells had a much higher increased propensity to generate new cells that failed to survive. So the new cells that were made just really were unhappy. And so as a result, they died. Now that, of course, can lead to a lot of problems in the brain because you have abnormal cell death, which can result in fewer neurons being made. The second problem that we observed was that they had an increased likelihood of not making enough new precursors. And as I mentioned earlier, these progenitors are a type of stem cell, and stem cells, by definition, need to make a differentiated cell, but they also need to make more of themselves. And if they're not making enough of themselves over time, you're going to basically run out of the maternal cell. You're going to run out of that neuronal precursor. And so the end result of that is that you won't make enough neurons. So this is we think that this is important in the context of disease because, and in particular in the context of a disease called microcephaly. Microcephaly is associated, is defined by brains that are significantly smaller in size. And then individuals who exhibit microcephaly can have varying degrees of intellectual disability. And a number of the genes that are mutated in microcephaly, actually the vast majority, are associated with mitosis.
I remember, Debbie, reading, I think this was in Latin America, there was some sort of parasite that results in this, right?
Yeah, so you're probably thinking about the Zika. There was an outbreak several years ago. Now I guess it's about 4 years ago in Brazil, where when pregnant women were infected with Zika virus, there was a significant increased likelihood, I think 5%, of pregnancies, depending on where you look, were resulting in babies that were born microcephalic. And so actually one of the things that we studied in the lab for some time was whether Zika itself was affecting mitosis.
Mitosis, yeah. And so one reason here, if I understand this correctly, is sort of early and programmed cell death, right? And so they don't have a chance to develop into neurons. Is that the right way to think about it?
Yeah, I mean, it is. If basically you make new cells that even begin to have the early features of a neuron, but those cells undergo cell death, the end result is that you're going to have fewer cells in a brain, and that's one clear explanation for microcephaly is abnormal or excessive cell death. So, and that can happen at various stages of development. There's also microcephalies that can result from agenesis or abnormal cell death that happens after birth.
Yeah, so I don't know anything about this, Debbie. So the size of the brain, it just seems weird that some mechanics like the Zika virus infection, obviously it affects the neuron production, but how does it sort of turn back to the size of the brain ultimately?
So probably the best way to think of it is that the brain has the potential to be a certain size, and if you have a genetic mutation or something like a virus that disrupts the ability to make all of the cells that brain needed, the brain is ultimately going to be smaller in size. And something like microcephaly can result from not only excessive cells dying, but also just the inability to make enough of the cells that are needed. And both of those aspects can give rise to microcephaly. Likewise, there's also macrocephaly. In fact, macrocephaly has been associated with autism, and that is the definition of macrocephaly is a larger brain. And there are some studies which have shown that in some patients, some autistic patients, there are actually more neurons made. So it's the reverse of microcephaly.
So anytime we see sort of unexpected brain size, this is one of the causes. You talk about cell death but not differentiation is P53 dependent. What is P53?
Oh, well, P53 is actually best known for its role in cancer. And it's a master regulator transcription factor that controls a whole number of processes, including the ability of cells to divide and the ability of cells to survive. So if a brain is lacking p53, what ends up happening is some of the triggers for cell death are missing. And so you could have, for example, a brain that was prone to exhibit excessive death of neurons. And if you remove p53, those neurons now won't die. And in fact, in some cases, that can be in a model organism a way to rescue the microcephaly.
So p53 sort of impedes cell death.
So p53 is going to be upstream of promoting cell death, actually. So, but p53 is referred to as a guardian of the cell. It's not going to always be needed, and so. In something like cancer, if you have excessive damage to the cells, like say from UV or damage to the DNA, something like p53 actually gets activated and it says, we have a problem here. We need to get rid of these cells. And so that's how it operates in a cancer, where it could operate in like a cancer context. And the same actually happens in the developing brain. We know of these systems where if there's excessive damage that occurs, p53 will get activated.
Right. Okay. And so p53 is sort of good when you have cancer type phenomenon. You need to kill them off. But if it is in the brain, perhaps in excess quantities, that is when. So removing p53 from the brain, as you mentioned, reduces the chance of tissue.
So removing p53, which is kind of, it's a very artificial experiment, but we and others have observed that if you have a brain that's prone to have excessive cell death, and as a result, the brains are going to be smaller. If you remove p53, in many cases, that can be sufficient to rescue the brain size. Now, I do want to say there's been some really interesting studies that have suggested that this is, and not surprisingly, that this also can create its own problems. So right. If you remove p53, you're now allowing cells that the brain should have disposed of because they had problems to stick around, and that can result in a whole nother host of problems.
Or could be very directly connected to cancer again.
Yes, exactly. It could predispose someone to cancer. So it's certainly by no means a treatment, but it's kind of a way to learn more about cellular processes and molecular targets that could be interrogated for potential ways of ameliorating something like microcephaly.
I want to touch on one other paper, Debbie. You're talking about pathogenic mutations that impair RNA. Mm-hmm. And related issues in the fetal cortical development. You want to talk a bit about that?
Sure. We had a recent study that we began studying a gene that regulates RNA, and it's a gene called DDX3X. And we became interested in this gene because at the time there was evidence that this gene was mutated. Had at least about 36 known mutations in it that caused intellectual disability. And these mutations arose de novo, meaning they were not inherited from mom to child. Rather, at the very earliest stages of development, they arose. And so what we decided to do was to really use our model systems to better understand how these mutations may be acting on brain development. And so there's two types of mutations that are associated with this gene. The first type is a mutation that is predicted to be a loss of function, and what that means is it eliminates at least a large fraction of the DDX3X gene. The second type of mutation is what's referred to as a missense mutation. And this doesn't necessarily get rid of the good gene, but it actually changes the function of it by just modifying certain features of the gene. And so we were able to deduce that if we replicated that loss of function by eliminating the gene during early development, it impaired the brain's ability to make neurons. And then we worked together with a clinical collaborator, Elliot Shearer, who's at UCSF, and he discovered through as a clinician that there were well over 100 now mutations in this gene that cause intellectual disability. So this tells us that this is a gene that's really, really important, and there is important for brain development, and that by looking at these individual mutations, we can get a better handle on how these different mutations can specifically impair neuron production and neuron function.
Right. Yeah. I mean, every time you think about this, it's a miracle the brain actually works.
Yeah, it is true. There's lots of safeguards, in fact, that the brain has for repair. There's DNA repair mechanisms in place to ensure that we don't have mutations, that are replicated. But you're absolutely correct. There's a lot of we focus a lot on the things that can go wrong, and that's mostly because in science that can give us a handle on important processes, but obviously there's a lot that goes right.
Yeah, and it's sort of very finely balanced, right? It's sort of balanced on a knife edge in some ways, that if you take it one way or the other, slightly off base, it just, so many different things could happen to it, right? And that's why it's a miracle.
Yep.
Yeah. Only a small percentage of the population actually has problems or show problems.
Well, there's a great colleague in this field, Chris Walsh, who's been doing some really elegant work to better understand to what extent our brains actually have mutations. And he's discovering that, all of us are walking around with various mutations, but many of those mutations are just going to be innocuous. So you can have changes in your DNA that really, in the end, don't cause problems. But if they happen to land in an important gene, that's where it can result in diseases.
Right, or we could get a beneficial mutation and everybody's IQ goes to
5. There you go. That'd be great.
So you have done a lot of work in this area, Debbie, in terms of developing brain. So if you look forward, in conclusion, if you look forward 5 years, where do you think we will make, sort of the most impactful discovery in this area?
Well, I think there's a lot of technology that has begun to enable a much more comprehensive understanding of brain development. We now have technologies where we can investigate human cells using what are referred to as organoids, where you can make a structure that resembles early developing brain, and you can use that to understand important features. But we also have better microscopy methods that we can use to understand not just these features in a tissue, but how they all interconnect with each other. And then on top of that, there is just amazing genomic technologies that are now allowing us to combine all of these together to understand not only gene expression, but also how gene expression is integrated across different cell types. And then the additional layer I would say that I'm particularly excited about is being able to then ask about, levels of conservation between developing human brains and other species, because we can use that information to better understand what are the features that make us uniquely human, but also to better gain a sense of what causes diseases.
Just very quickly, I wondered, do we have any data to show if the human brain has changed over time? And by over time, I mean thousands of years.
I know of, that we know about the genomes of Neanderthals and their relatives relative to Homo sapiens. And there are differences in the genomes in terms of how the brains have changed. I think that is an area that is one that we still don't know enough about. But it's really interesting when you think about it come up from a bigger perspective. How have changes in our abilities influenced our brain's evolution, our technologies, etc.
Right. Yeah. I know that very tactically, our IQs have increased marginally. I wondered if it's a measurement problem or it's just the work that we do with it that's also sort of forces it to go up the stream. But I think architecturally there hasn't been much difference, right? I mean, Homo sapiens have been around only for 50,000 years. I think it's just a short time to really have a huge difference.
Yeah, I mean, I think you're right. There are what we might refer to as subtle but important differences. But the anatomical and cellular and even molecular basis for those differences is I think one that is going to keep developmental biologists and evolutionary biologists busy for many years to come.
It's good job security.
Yeah, that's right.
This is one of those areas that every time you look at it, you get some new information, but then you find there is so much more behind it. And so, it keeps teasing in terms of knowledge, but it seems like we are not getting any closer.
Well, as I was highlighting some of these technological developments, like the ability to reconstruct human tissue in a dish, the ability to interrogate gene expression, many of these can be applied across different species and even across different species of humans potentially. To get better, to begin to, understand some of these questions.
Yeah. Excellent. Yeah, this has been great, Debbie. Thanks so much for spending time.
Yeah, thank you so much for the invitation. This was a lot of fun.
Yeah, and good luck with this research.
Yeah, thank you. Okay, bye.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.